Hello, this is Maria Grant. Dr. Alan Taylor and I have put together a series of slides that discuss the role of the gut microbiome and how it may play a role in the pathogenesis of age-related macular degeneration. Eubiosis is the condition in which the gut is perfectly in balance. In contrast, dysbiosis occurs when the gut microbial community is out of balance. This occurs in conditions such as obesity, metabolic syndrome, and diabetes. The gut becomes leaky. There's disruption of the epithelial barrier. Microbes and microbial products enter the circulation where they can exacerbate inflammation, as well as result in disruption of the blood-brain barrier and blood-retinal barrier. The term diversity is referred to as the degree or amount of variation in a microbial community. There are several types, primarily alpha diversity and beta diversity. Alpha diversity refers to the amount of variation within a sample, whereas beta diversity refers to the variation between samples. The analysis of the microbiome begins with an assessment of the actual bacteria. This is done using 16S ribosomal RNA sequences for the bacteria. The term OTU, or Operational Taxonomic Unit, is a way to group the bacteria by the DNA sequence. The other term of importance is abundance. Specifically, relative abundance refers to how common or rare a particular OTU is relative to other OTUs in the same community. Richness refers to the number of OTUs present in the community, and it's believed that the richer the community, the more robust and the healthier the host. Dr. Mike Sapia's lab presented an elegant study in which they described the role of high-fat diet on modulation of the gut microbiome. It's well appreciated that after smoking, obesity is the next highest risk factor for development of AMD. In this little schematic, we show the high points of the paper. The group compared regular chow-fed mice to high-fat diet mice. They found that the diet impacted the microbes found within the intestine and resulted in dysbiosis. The dysbiosis resulted in an increase in intestinal permeability with the generation of higher levels of LPS and other pathogen-associated molecular pattern molecules, or PAMPs. PAMPs then activate receptors. The receptors are called pattern recognition receptors that are on the surface of innate immune cells. PAMPs trigger the activation of these receptors, and these receptors in turn stimulate the release of large numbers of cytokines into the circulation, increasing inflammation and provoking or worsening CNV formation. I'm going to spend a minute showing you some data from this paper that supports the significance of the microbiome in the pathogenesis of these changes that were observed. In this experiment, you can see on the timeline 6, 9, 11, and 13 weeks, the animals are placed either on a regular diet or high-fat diet. They're given neomycin to eliminate their intrinsic microbiome, and then they're placed on different diets. Regular diet plus antibiotics, high-fat diet plus antibiotics. Once again, the use of the antibiotics is to minimize any changes in the microbiome. Then, at 11 weeks, the animals undergo laser burns, and at 13 weeks, they're sacrificed. If you look at this panel here, you can see confocal images of the CNV lesions. These are representative lesions, but you can clearly see that the high-fat diet-fed mice lesions are higher than those of the regular diet mice. And the high-fat diet mice given antibiotics is smaller than the high-fat diet mice that are not given antibiotics. If you look here at this image, you can see the quantitation of the actual areas of the CNV lesions, and this supports what we just discussed. The high-fat diet mice had higher, larger lesions. And this was reduced, essentially normalized by use of antibiotics. This is suggestive that there indeed an influence of the microbiome on this effect. If you look at two populations that are the major phyla of the microbiome, they are Bacteroides and Firmicutes. In the regular diet-fed mice, there's a predominance of Bacteroides and a lower level of Firmicutes, and this ratio is consistent in healthy animals. Interestingly, in the high-fat diet-fed animals, Firmicutes becomes a predominant phyla with a marked reduction of Bacteroides. Interestingly, the high-fat diet-fed mice given antibiotics had a microbiome similar to the regular diet mice. And the reduced levels of Firmicutes is what is typically seen. When you have a high-fat diet mouse receiving the high-fat diet transplant, there's an imbalance here with a much higher predominance of Firmicutes and less Bacteroides in red. Interestingly, when these animals received transplantation from mice that received a regular diet, this balance was corrected and it was much closer to what is seen under normal conditions. These results support the impact of how important the microbiome is in the lesion size development, and also that fecal transplantation can correct the dysfunction. Work from Alan Taylor's lab show the importance of glycemic index. We just discussed the importance of high fat, and here we're now considering high glucose. So, if in his elegant studies that were published in PNAS, he examined the impact of different types of diets and switching from high glycemic index to low glycemic index. I'd like to direct your attention to one figure from his paper where he looked at outer nuclear layer thickness. As you can see here, the low glycemic shown here in blue and the initials LG, this is the typical thickness pattern from the optic nerve head. The high glycemic index animals had a marked reduced thickness. However, the high glycemic index animals that were then switched to low glycemic index diet were able to have restoration of their outer nuclear layer thickness towards normal. In some lovely immunohistochemistry studies, Dr. Taylor's group examined the effects of high glycemic index diet on the retina. As shown in the lower panels, one sees subretinal deposits, vacuoles in the RPE atrophy, as well as hypopigmentation. Interestingly, the animals that were on the high glycemic index and switched to the low glycemic index had marked improvement of their retinal images. He also did a very detailed retinal damage score, and this is a summary of the results. Low glycemic index, healthy retina. High glycemic index, a retina demonstrating damage. And the animals that were switched from being on high glycemic index to low glycemic index diet had restoration or improvement of their retinal damage score. Here is the detailed timeline that describes the events that I just discussed. We have the animals that received the high glycemic index diet. And you can see that over time, 18 months and 24 months, at this point the mice are really aged. And here they're in midlife. There's progressive increase in the damage. Interestingly, the animals that were on the low glycemic diet did very well until about 18 months, and then some injury was detected by 24 months. However, the markedly significant aspects of this study was the observation that if you took the animals on the high glycemic index and switched them to the low glycemic index, it normalized their changes towards those just seen within the aged normal cohort that was on the low glycemic index diet. Dr. Taylor's group went on to show ultrastructural changes. They examined lipofuscin accumulation. As one can see on the left panels, there's electron micrographs that show the loss of basal infolds, vacuoles, and formation of large basal laminar deposits along the membranous with Dr. Taylor's group was able to show that the high glycemic diet induced ultrastructural changes, including lipofuscin accumulation in the RPE. These studies were in mice that were 24 months old. On the left, you can see electron micrographs that show the loss of basal infoldings, as well as vacuoles, as well as the formation of large basal or laminar deposits along with membranous debris, and the accumulation of lipofuscin, phagosomes, and lipid deposits in the animals that were on the high glycemic index diet. The number of lipofuscin granules is linearly related to the extent of retinal degeneration, as shown here in this graph. Autofluorescence is shown in yellow, and most likely represents lipofuscin granules that are dramatically increased in the high glycemic index animals. Interestingly, when the high glycemic animals were switched to the low glycemic diet, there was a reduction in lipofuscin granules. Here we see the quantitation of these interesting studies, and also not only for the lipofuscin granules, but also for the phagosome numbers were markedly increased in the high glycemic index diet and were restored almost to normal in the crossover study. My lab has been interested in the role of the microbiome in diabetes, and our interest began with studies using intermittent fasting. Intermittent fasting has gained much popularity in the recent years. And one of the benefits is to restore circadian dysregulation towards normal. Intermittent fasting also improves energy sensing pathways such as AMPK, AKT, and others. It's also appreciated that microbial-derived factors modulate many pathways in longevity, further implicating the microbiome as being important in healthy aging. We wanted to see if intermittent fasting could prevent the development of diabetic retinopathy. Indeed, we found that 7 months of intermittent fasting prevented acellular capillaries from forming, prevented the inflammatory changes within the retina. However, to determine the mechanism by which this was occurring, we examined the microbiome, knowing that it could be impacted by this fasting regimen. We found that the bacteria that were increased under these conditions were those that generated secondary bile acids. In this slide, we can see the 4 cohorts of the study on the bottom, green being the control on ad libitum feeding, diabetic on ad libitum feeding, control on IF, and diabetic on the IF regimen. What we found was that one particular secondary bile acid, TUDCA, or tauroxodeoxycholate, was increased in the cohort on the intermittent fasting regimen. This particular bile acid is known to be neuroprotective. The receptor for TUDCA is TGR5. TGR5 is a G protein coupled receptor and is also a nuclear hormone receptor. We examined the expression of TGR5 within the retina. As you can see on the images here, we used immunohistochemistry and detected TGR5 by the green staining. Nu-N, or the ganglion cells, are in red, and DAPI is in blue. The ganglion cell layer is shown here, as well as the inner and outer nuclear layers. We did not detect any significant difference in expression of TGR5 in any of the 4 cohorts. However, we examined downstream signaling for TGR5 by examining the expression of TNF-alpha mRNA. In the diabetic ad libitum cohort, there's increased mRNA expression for TNF. And this was reduced in animals on the intermittent fasting protocol, suggesting that TGR5 activation was increased in the diabetic cohort on the IF regimen. In summary, our study showed that intermittent fasting is able to prevent diabetic retinopathy, and the mechanism is in part due to manipulation of the microbiome. We believe that intermittent fasting restructured the microbiome in favor of bacteria that generated secondary bile acids such as TUDCA. TUDCA is known for its neuroprotective effects. TUDCA was able to activate TGR5 receptors on the retina, specifically the ganglion cells, resulting in reduction in TNF production and also reduction in inflammation. Recently, we became interested in understanding the role of the microbiome and the renin-angiotensin axis. The classical RAS consists of angiotensinogen that is degraded by renin, derived from the kidney, into angiotensin 1. Angiotensin 1 is then converted by ACE to angiotensin 2, which then can mediate its effects through either the AT1 or the AT2 receptor. In general, the AT1 receptor is known for the deleterious effects of angiotensin 2. It's mediating vasoconstriction, fibrosis, inflammation, and cell proliferation. The deleterious arm of the renin-angiotensin system is counterbalanced by the protective arm. Key components of the protective arm is angiotensin-converting enzyme 2, shown here in the box, which takes angiotensin 1 and converts it to angiotensin 1,9 and angiotensin 2 to angiotensin 1,7. Ang-1,7 can then activate its receptor, the MAS receptor, to have many beneficial effects on the vasculature as well as other tissues, including vasodilatation, antifibrosis, anti-inflammation, and anti-proliferation. The RAS axis has been central in the pathogenesis of many disease states, where it's believed that the deleterious arm is outweighed in its effects compared to the beneficial arm. Atherosclerosis, hypertension, cardiac disease, kidney disease, and diabetes have all been associated with activation of the renin-angiotensin system, specifically ACE1, AT1. We examined the role of the renin-angiotensin system on the gut microbiome. We did this by examining mice that were deficient in ACE2. We studied wild-type Akita, the ACE2 knockout mice, as well as a double knockout in which the Akita mice were also deficient in ACE2. When we examined the microbiome, we noted that there was difference in the beta diversity of these cohorts, suggesting that each cohort of mice had a distinct microbiome from the other cohorts. When we examined a heat map that described the actual most prominent bacteria in each group, we found that Bifidobacterium animalis was very high in the double knockouts. This cohort of mice were then examined further. We next examined the metatranscriptomic profile of the gut microbiome in all the cohorts. The most interesting data was in the double knockout, where peptidoglycan biosynthesis was markedly increased. The bacteria responsible for this gene signature was Bifidobacterium animalis. We then wanted to evaluate whether there was significant dysbiosis. One would predict that there would be, the combination of diabetes as well as the loss of ACE2. We evaluated both the epithelial and the endothelial barrier. In the interest of time, I'm going to just show a little bit of the data from the endothelial study. We selected a protein called plasma lemma vesicle-associated protein 1, or PV1. This protein is selectively expressed in microvascular endothelial cells. Interestingly, this protein is expressed in higher levels when there's disruption of barrier characteristics, or when there's barrier disruption, there's higher levels of PV1. As you can see on the immunohistochemistry of this particular study, there's wild type, the Akita, the ACE2, and the double knockout. When you perform semi-quantitative analysis, we can certainly see that the double knockout has the highest expression, suggesting that this cohort has the leakiest gut vascular barrier. While PV1 goes up in expression with increased leakage, V-cadherin goes down, and this is classically seen whether it's the retina or the gut. There's decreased expression of V-cadherin suggests more loss or greater disruption of the endothelial barrier characteristics. As one would expect, when the barrier gets disrupted, there's increased leakage of peptidoglycan into the circulation of these mice, and this is shown here in this particular slide. We then examined the mechanism to understand what was happening. How was the disruption of the gut barrier occurring? While we anticipated that there would be changes in mucin production, while we anticipated that there would be changes in inflammation, suggesting there was more leakage of we expected that there would be more inflammatory cells in the diabetic intestine. Surprisingly, what we found was that there was a loss of vascular reparative cells. We then did a considerable number of studies to confirm this using flow cytometry. However, I'm just going to show a little bit of the data of this study to emphasize that the bone marrow communicates with the gut and that these protective cells were reduced in the diabetics and particularly reduced in the double knockouts. When we replaced the loss of these cells by giving them 3 times over a 6-day period, we were able to improve and restore barrier characteristics. As shown here, this is control. These are animals that were given saline, and here are the animals that received the stem cells or circulating angiogenic cells. You can see between these two there was a reduction in PV1, suggesting that the administration of circulating angiogenic cells improved barrier characteristics. This is shown here for the jejunum and also for the ileum. Similarly, if you look at the controls which were given saline compared to the animals given circulating angiogenic cells via intraperitoneal injection, the animals that received the circulating angiogenic cells had an increase in their V-cadherin expression, suggesting that the cells were able to restore and improve barrier characteristics. This is summarized in this schematic. In diabetes, as well as in aging, it is well known that there's a reduction in vascular repair and that the bone marrow becomes dysfunctional. While we study diabetes, it would not be difficult to extrapolate some of these findings to the aging population, where we know there's increased damage, yet there's reduced repair. What we were able to show is that by giving circulating angiogenic cells or vascular reparative cells, we were able to improve the integrity of the gut vascular barrier in the diabetic animals. This in turn resulted in less damage to the retina and an intact blood-retinal barrier. In the interest of time, I didn't expand on all this data. However, we were able to show that indeed gut vascular barrier and blood-retinal barrier had parallel characteristics. When one was intact, so was the retina. And this is in a recent publication in Circulation Research. The last component of this tutorial is to emphasize the potential therapeutic use of bacteria. Dr. Kyu-Hong Lee, University of Florida, genetically modified bacteria to produce ACE2. The bacteria she used was Lactobacillus paracasea, and they served as a live vector for the oral delivery of human ACE2. This study was recently published in Molecular Therapy, and I'd just like to point out two parts of this paper. One was that by administering the bacteria, here shown as ACE2LP versus saline, that she was able to increase serum ACE2 activity as well as ACE2 activity in the kidney and lung and retina. The expression of human ACE2 by the bacteria had beneficial effects when they were given to the Akita mouse. Here we can see the different cohorts. Control, the Akita having increased numbers of acellular capillaries, and then the Akita mouse receiving the wild-type bacteria, and the Akita mice that received the ACE2 expressing bacteria. In addition to improving retinopathy, she confirmed that there was less TNF, IL-1 beta, ICAM, and MCP1 in the retinas of the ACE2 she was able to show that the ACE2 expressing bacteria, when given to the Akita mice, reduced the number of acellular capillaries, and when examining the retina for expression of pro-inflammatory cytokines, the Akita mice receiving the ACE2 expressing bacteria had less TNF-alpha expression, less IL-1 expression, as well as reduced levels of ICAM and MCP1. In conclusion, there is considerable data to show that high-fat diet can have a deleterious effect on the size of CNV lesions in rodent models. Similarly, a high glycemic diet can have a deleterious effect on the RPE cells. Intermittent fasting can be protective by restructuring the microbiome, favoring the generation of neuroprotective bile acids that can protect the retina in diabetic animals. Stem cells can serve to enhance barrier function, resulting in the reduction of the release of microbial peptides into the circulation. These peptides can activate the endothelium and cause further barrier disruption in the retina. They can also activate circulating cells that can, in turn, enter the retina in an activated state and cause damage. Finally, the future may hold the use of genetically modified bacteria to express therapeutic proteins such as ACE2. Thank you for your attention.